Hi, welcome to this PowerPoint on muscle contraction. Muscle contraction is an essential function of the body and is vital in everyday activities as well as sport. Muscle contractions occur very quickly and react to stimuli which is picked up by the sensory organ, which is often the eyes in sport, and travel down sensory neurons to the central nervous system. Next, it travels down motor neurons where it reaches a synapse called the neuromuscular junction, which allows the motor neuron to transmit a signal to the muscle fibre. This then leads to the sliding filament theory, which involves the proteins actin and myosin. This overall process causes the muscle to contract and move. Muscle contractions happens throughout many sports. An application of muscle contraction due to a stimuli is catching a ball, whether that's a basketball, a cricket ball, as shown above. However, the example I'm going to use is of catching a rugby ball, as you can see Chris Robshaw doing in the picture below. So, when the ball is thrown towards a person, this is known as a stimulus, and this is detected by the sensory organ, or receptors, which in this case is the eyes. This causes the sensory neurons to send an impulse, which is an electrical signal, to the brain, which is part of the central nervous system, where it's passed on to a motor neuron, which carries the impulse down to the effector muscle, which carries the impulse to the effector muscles, which in this case will be many different muscles in the arm in order for them to contract. Motor neurons consist of three main parts: dendrites, cell body, and axon. Dendrites collect the electrical signal and carries it to the cell body, which only contains one nucleus, and transfers it down the axon, which passes electrical signals to dendrites of another cell or onto an effector cell. However, before the impulse can reach the muscle for the muscle to contract, it has a gap to overcome called the neuromuscular junction. This synapse is where a motor neuron communicates with the skeletal muscle fibre. At the end of the motor neuron, an axon terminal, which are enlarged ends that contain vesicles of the neurotransmitter. These vesicles contain a chemical called acetylcholine, and this is secreted across the synaptic cleft and bonds to receptors on the motor end plate of the muscle fibre. If enough is secreted and electrical charge is high enough, then the muscle action potential is created. Neurons stimulate sarcolemma by generating an action potential from an electrical impulse. The cleft and motor end plate contain acetylcholine esterase, which breaks down ACH as it is passed on and when it arrives. The sliding filament involves two main proteins. The sliding filament theory involves myofibrils, which contain two main proteins: myosin, which is a thick filament, and actin, which is a thin filament. Myosin is thick with a tail and a rounded head that attaches to actin binding sites during contraction. They are also much larger than actin. Actin is thin and twisted filament and has specific sites for myosin to bind to. However, at rest, these sites are covered by tropomyosin, which is held in place by troponin. At rest, myosin is ready to contract, however they can't contract as the actin is blocked by the troponin and the tropomyosin, which were mentioned in the previous slide. Muscle cells are composed of actin and myosin molecules in a series. A basic unit of these in the muscle cell is known as the sarcomere. When the impulse reaches the neuromuscular junction and enough acetylcholine is secreted, the impulse travels along T-tubules and into the sarcoplasmic reticulum, which releases the calcium ions. The sarcoplasmic reticulum is a specialized smooth endoplasmic reticulum that contains high concentrations of calcium ions. This is stimulated from the impulse from the T-tubules to release the Ca+ ions for muscle contraction to occur in the fibers. Calcium ions, which are stored in the skeleton and released into the bloodstream under very controlled conditions, bind to the troponin and causes it to change shape. This then pulls the tropomyosin away as well, exposing the actin binding site, making a cross bridge. Next, the ATP is broken down in an exothermic reaction, which releases the energy needed for the power stroke, which actually causes the sarcomere to shorten. This is repeated along the whole of the myofibril, so the muscle can contract. The The sarcomere shortens as the Z lines contract towards the M line. Now the Z lines are thin filaments at both ends of a sarcomere. Another protein connects to the Z line to the thick filaments to maintain their alignment. The M line is made of connections between the thick filaments. Once one power stroke has occurred, the myosin takes in more ATP and is released, ready to make another cross bridge. This means the myosin molecules act like a ratchet, while the actin molecules form passive filaments that transmit the force generated by the myosin to the end of the muscle tissue. The mechanism of the sliding filament theory involves the myosin progressing along an actin filament, constantly binding, ratcheting, and then letting go. This process allows muscles to contract. ATPase is an enzyme that catalyzes the reversible reaction, which is shown on the bottom of the screen here, enabling a quick supply of energy for muscle contraction. In order to maintain muscle contraction, a stimulus is needed, and also a continuous supply of ATP and Ca+ ions. Within a sarcomere lies A bands, I bands, and H zones. A bands contain thick filaments, I bands contain thin filaments, and H zone is the distance between two adjacent actin filaments. All are demonstrated in the figure, just on the right of your screen. At rest, all three are at maximum width within the sarcomere. However, when muscles contract, I bands and H bands shorten, and when fully contracted, H zones disappear and I bands become very narrow. On the other hand, A bands stay the same width. Once the stimuli stops, the electrical stimulation ends, which causes the sarcoplasmic reticulum to recapture Ca2+ ions, and troponin and myosin revert back to their original positions, which is when they're covering the actin binder sites, which means no more movement can occur. Now there are four main types of muscle contractions, two of which are isotonic, so you have concentric and eccentric, and also you have isometric and isokinetic contractions. Concentric contraction is a type of isotonic contraction and is the main type of muscle contraction and also the most common. It occurs when the muscle shortens at both ends and they become closer together. This is a type of muscle activation that increases tension on a muscle as it shortens. This contraction is best demonstrated when doing a bicep curl, as when lifting the dumbbell towards your body, as your biceps brachii will contract. This movement also occurs frequently across many other sports, such as competitive weightlifting and running, particularly when uphill. The other isotonic contraction was eccentric contraction, which is where the muscle increases in length while still producing tension. The two muscle ends move further apart. Eccentric contraction is the contraction that opposes gravity and means gravity can't occur at full force. It's the opposite of concentric, and a simple example, as shown on the left, is the lowering of a dumbbell in a bicep curl, as it stops the arm dropping straight to the floor. A more obscure example of this is the tibialis anterior muscle, which contracts eccentrically when running to keep control of your foot and means your foot cushions the floor. Next is isometric contraction. This is where the muscle length does not change and the joint angle does not alter. As there's no movement, isometric exercises are done in static positions. However, the muscle fibers still fire. A simple example is pushing against a brick wall. This movement can be used for rehabilitation as well as general strengthening of the muscle. Many sports also require it at a high level and require a high level of static muscle strength. For example, gymnastics, rock climbing and skiing. In particular, gymnastics, as you can see on the left, as you need to hold a variety of positions. However, these positions involve great strength. It's just there's no movement in those positions, in the muscle or the surrounding joint. And finally, we have isokinetic contractions. These are the least common and are similar to isotonic contractions in that the muscle changes length during contraction. However, it differs in that it produces contractions of constant speed. To measure this muscle contraction, a special piece of equipment called an isokinetic dynamometer is required. Isokinetic movements are a lot more rare. However, one example, as you can see in the image on the screen, is breaststroke, where in swimming, where water resistance is a constant. And that's it. Thank you for watching.